everyone, we are the group 2 and we'll be discussing about protein. But before that, let me give you a brief context about protein. Protein are the essential molecules made up of amino acids. They perform various functions in the body such as building structure, catalyzing reactions, and transporting substances. Now, let's dive deeper to understand more about protein. Protein are the macromolecules that play an integral role in the proper tackling of cells. Proteins are large molecules essential for cell function, participating nearly in every cellular process. Their diverse roles are crucial for cell survival and proper operation. Proteins are built up of linear polymer of amino acids. Proteins are chains of amino acids linked together to form a polypeptide chains. The specific sequence, unique 3D structure, and function. Proteins play an operational or mechanical role in the cell cytoskeleton. Proteins are key components of the cell cytoskeleton, providing structural support, enabling cell movement, and facilitating transport within the cells. This framework is essential for maintaining cell shape and function.
Proteins are natural polymers. Proteins are large molecules that play crucial roles in biological systems, such as in cell structure, function, and regulation. A polymer is a long chain made up of smaller, repeating units. In the case of proteins, the repeating units are amino acids. They are very large molecules that are critical for the functions of the human body. Proteins are very large molecules because they can consist of hundreds or thousands of amino acids joined together in specific sequences. Proteins are critical for the functions of the human body because they perform many essential tasks. These include acting as enzymes, structural components, signaling molecules, and antibodies to protect against disease. They are made from the leakage of monomers called amino acids. Proteins are made by linking together small molecules called monomers. The monomers in this case are amino acids, which are organic compounds that contain both an amino group and a carboxyl group.
Amino acids are the monomers that make up proteins. Each amino acid has the same fundamental structure, which consists of a central carbon atom, also known as the alpha carbon, bonded to an amino group, a carboxyl group, and to a hydrogen atom. In the aqueous environment of the cell, both amino group and carboxyl group are ionized under physiological conditions and so have the structures ammonia and carboxylic acid. Every amino acid also has another atom or group of atoms bonded to the central atom known as the R group. These R group are side chains, gives each amino acid proteins specific characteristics including size, polarity, and pH. The chemical composition of the side chain determines the characteristics of the amino acid. Amino acids such as valine, methionine, and alanine are nonpolar or hydrophobic, while amino acids such as serine, threonine, and cysteine are polar or hydrophilic. The side chains of lysine and arginine are positively charged, so these amino acids are also known as basic or high pH amino acids. Proline is an exemption to the standard structure of an amino acid because its R group is linked to the amino group, forming a ring-like structure.
Structure of amino acids. Each amino acid has a central carbon atom with 4 covalent bonds. Central carbon atom is also known as alpha carbon. The name of amino acid is represented by a single uppercase letter or a 3-letter abbreviation. The 4 covalent bonds are 1 bond with hydrogen atom, 1 bond with amino group, 1 bond with carboxyl group, 1 bond with a unique side chain that determines the amino acid specific properties. Those all amino acids have a central carbon with 4 covalent bonds which is essential to their structures.
Atoms, ions, and molecules. As you can see, there's an example below, and we see three important chemical groups. The first group is the anomide group. This group contains nitrogen and hydrogen, letter N and H. It is commonly found in amino acid, which are the building blocks of proteins. The second group is the functional group, letter R. The R here represents a variable side chain that can change in structure. This determines the specific properties of a molecule, such as the different types of amino acid. And lastly, the carboxyl group. This group contains carbon, oxygen, and hydrogen, letter C, O, and H. It's a defining feature of organic acid like amino acid and fatty acid. And together, this group help form the structure of biological molecules like proteins, which are essential for life.
There are 20 standard amino acids that are the building blocks of proteins found in the human body. These amino acids can be classified into different groups based on the chemical properties of their side chains. First is the nonpolar or hydrophobic amino acids. These amino acids have the side chains that are nonpolar and hydrophobic, meaning they are repelled by water. They tend to be found in the interior of proteins, away from the aqueous environment. These are the alanine, glycine, isoleucine, leucine, methionine, phenylalanine, proline, valine, and tryptophan. Second is the polar or hydrophilic amino acids. These amino acids have side chains that are polar and hydrophilic, meaning they are attracted to water. They are often found on the surface of proteins, interacting with the aqueous environment. These are the asparagine, glutamine, serine, threonine, tyrosine, cysteine, and histidine. Next is the charged amino acids. These amino acids have side chains that carry a net charge at neutral pH. They are strongly hydrophilic and play a crucial role in protein structure and function. These are the aspartic acid, glutamic acid, lysine, and arginine. And lastly, special cases, which are the asparagine or aspartic acid, glutamine or glutamic acid. These special cases represent stances where it is difficult to distinguish between two closely related amino acids.
Proteins are based on the number of amino acids in a chain. First one is peptides. Peptides are short chains of amino acid linked together by peptide bonds. These are fewer than 50 amino acids, and although they are small, peptides can have significant biological activities. For example, some hormones like insulin are peptides. Second is the peptides. The peptides are the simplest form of peptides consisting of two amino acids. These are formed through a condensation reaction where two amino acids combine, releasing a molecule of water and forming a peptide bond. An example of these are carnosine, which are made of beta-alanine and histidine. These are often found in protein breakdown products and can play various roles in metabolism. The third one is tripeptides. These consist of three amino acids linked by peptide bonds. They are slightly more complex than dipeptides. They can exhibit unique properties and functions. An example of this is glutathione. The fourth one is polypeptides. Polypeptides are longer chains of amino acids, typically containing more than 10 amino acids. They can fold into specific three-dimensional shapes, which are essential for their biological function. The sequence and number of amino acids determine the polypeptide's properties. The last one is proteins. Proteins are large, complex molecules made up of long chains of amino acids, typically more than 50 amino acids, and can range from 100 to 10,000 amino acids. Proteins can be composed of one or more polypeptide chains.
that fold into a unique shape, allowing them to interact with other molecules. In summary, the classification of proteins based on the number of amino acids highlights the diversity of protein structures and functions, from short peptides with specific roles to large proteins that perform complex functions. The arrangement and number of amino acids are fundamental to their biological activity.
Structural protein, found in the hair of mammals, tendons, and ligaments. Structural proteins are proteins that provide support and structure to cells and tissues. The image states that they are found in the hair of mammals, tendons, and ligaments. The examples of structural proteins include collagen, found in connective tissues like skin and bone, and keratin, found in hair, nails, and skin. Contractile proteins, proteins that provide muscular movement. This defines contractile proteins as proteins that provide muscular movement. This means they are responsible for the contraction and relaxations of muscle. Actin and myosin are the primary contractile proteins in muscle cells. Storage protein, specialized for storing amino acids. This describes storage proteins as being specialized for storing amino acids. Amino acids are the building blocks of proteins. Storage proteins act as reservoirs of amino acids which can be used later for protein synthesis. Examples of storage proteins include casein, found in milk, and ovalbumin, found in egg whites. These proteins provide a readily available source of amino acids for developing organism.
Transport proteins. Transport of substances. Transport proteins are responsible for the movement of substances across biological membranes. Signal protein. Coordination of an organism's body activities, also known as signaling proteins, play a crucial role in coordinating the activities of an organism's body. Enzymatic proteins. Selective acceleration of chemical reactions. It is also known as enzymes. It, it is a biological catalyst that accelerates chemical reactions in living organisms. Defensive proteins. Protection against antigens. Defensive proteins are responsible for protecting organisms against antigens, which are foreign substances that can trigger an immune response.
Levels of protein structure. First is primary protein structure, sequence of a change of anomic acid. Sample. This protein in red blood cell carries oxygens and is composed of 4 polypeptide chains. A single anomic acid can cause sickle cells anemia, affecting the red blood cells in shape and functions. Second is secondary protein structure. Local folding of the polypeptide chains in helix or sheets. Sample. These proteins that store oxygens in muscle tissue and some transmembrane proteins, which span cell membranes. And third is tertiary protein structure. Three-dimensional folding pattern of protein due to side change interactions. Sample. Responsible for regulating blood sugar levels consists of two polypeptide chains linked by demultified bonds. The tertiary structure of insulin is essential for its ability to bind to its receptor on target cells and trigger the uptake of glucose. And the last one is quaternary protein structure. Protein consisting of more than one anomic acid chains. Sample. This protein responsible for Oxygen transport in red blood cells consists of 4 polypeptide chains forming a tetrameric complex that binds 4 oxygen molecules simultaneously.
Primary structure is the unique sequence of amino acid in a polypeptide chain. Amino acids are attached together by covalent bond or peptide bond, and this is done through the process called protein biosynthesis. The primary structure of a protein is the unique linear sequence of amino acids in a polypeptide chain. Each protein's function and properties are determined by the precise order in which its amino acids are arranged. This sequence is encoded by the organism's DNA and is specific to each protein. In summary, the primary structure serves as the foundation of protein architecture. The unique sequence of amino acids joined by peptide bonds during protein biosynthesis dictates how the protein will fold and function in the body. Understanding this structure is essential for studying protein behavior, designing drugs, and investigating genetic diseases.
Secondary structure formed from local folding of polypeptide chain from protein. The secondary structure of a protein refers to the local folding of the polypeptide chain into specific shapes, primarily the alpha helix and beta pleated. Alpha helix structure. Helix is held in shape by hydrogen bond. Alpha helix structure. This is a right-handed spiral where the polypeptide chain coils around itself. The structure is stabilized by hydrogen bonds between the amide hydrogen and carbonyl oxygen atoms in the backbone of the chain. Beta-pleated structure are formed between hydrogen bonding atoms on the backbone of the polypeptide chain. Beta-pleated structure. This structure consists of extended strands of polypeptide chain that are aligned side by side. Hydrogen bonds form between the backbone atoms of adjacent strands, stabilizing the sheet-like arrangement. Both structures contribute to the overall shape and function of the protein.
Tertiary structure pertains to the overall 3D shape of a polypeptide by a pattern of folding. So, the tertiary structure of a protein is its final 3D shape after a chain of amino acids is made. It folds up in a specific way to form this shape. The way it folds is important because it helps the protein do its job. If it doesn't fold correctly, the protein might not work properly.
Quaternary structure, it is the arrangement of multiple folded protein. So quaternary structure, it is the protein that made up of 2 or more separate protein molecules that fold subunits that are joined together. Each subunits have its own function in our body. So quaternary structure contributes to the stability of the protein. So it is essential in our body. Example sa ating hemoglobin. So, yung hemoglobin ay yung mga red blood cells sa ating katawan na responsable sa pagdala ng oxygen sa ating katawan. So, syempre kung hindi magandang pagdala nito or structure nito sa ating katawan, pwede itong magka-problema na magdudulot ng sakit sa ating katawan. So, quaternary structure ay ang pinaka essential at sensitive sa lahat. So, kailangan nating alagaan ang ating katawan. So, in terms of pag-aalaga sa ating katawan, may mga pagkain na pwedeng pagkuhanan ng quaternary structure protein. Katulad ng sa meat, egg, and milk.